Welcome to Advanced Learning Tutoring. This is the biology playlist. Please like this video and subscribe to our channel. Communicable diseases are diseases that are able to be spread from person to person. An example of a communicable disease would be the cold or flu. The causes of communicable diseases are either viruses, bacteria, protists, or fungi. They are able to infect plants or animals and can be spread through direct contact by water droplets in the air or by air. Bacteria and viruses may reproduce rapidly inside the body. Bacteria produce toxins that damage tissues and make us feel ill, whereas viruses live and reproduce inside host cells, causing cell damage. Examples of viral diseases include measles, HIV, and tobacco mosaic virus. Measles symptoms include a fever, a high temperature, a red skin rash. Measles is spread by the inhalation of droplets from sneezes and coughs. HIV initially causes flu-like symptoms. HIV, the symptoms can be treated with antiretroviral drugs. This does not treat the overall viral infection, it just treats the symptoms. The virus attacks the immune system. Late-stage HIV, also known as AIDS, occurs when the immune system becomes so badly damaged it can no longer deal with other infections. HIV is spread by sexual contact or exchange of bodily fluids. Tobacco mosaic virus is a widespread plant pathogen affecting many species of plants. It gives a mosaic pattern on the leaves which affects plant growth due to lack of photosynthesis. Examples of bacterial diseases are salmonella and gonorrhea. Salmonella is a type of food poisoning that is spread by bacteria ingested in or on food. In the UK, poultry, such as chickens, is vaccinated against salmonella to control the spread. Fever, abdominal cramps, vomiting, and diarrhea are caused by the toxins secreted by the bacteria. Gonorrhea is a sexually transmitted disease. Symptoms include a thick yellow discharge from the vagina or penis, and also pain when urinating. It is caused by a bacterium and is treated with antibiotics. It is spread by sexual contact. The spread can be controlled by the use of barrier methods, such as condoms. An example of a fungal disease is rose black spot. This is a fungal disease that causes black spots to develop on the leaves, which often turn yellow and drop. It affects the growth of the plant as the lack of chlorophyll limits photosynthesis. It can be spread by water or wind from plant to plant. It can be treated with the use of fungicides or by removing and destroying the affected leaves. As the leaves have the black spots on them, this limits the number of chlorophyll in the chloroplasts as the leaves are less green. This is why it limits the rate of photosynthesis. Photosynthesis is the process that produces glucose. Glucose is used in respiration, which is the process of releasing energy. Therefore, less energy will be released for plant growth. An example of a protist disease is malaria. The malarial protist has a life cycle that includes the mosquito, the vector. Protist diseases will be carried by a vector. Malaria causes recurrent episodes of a fever and can be fatal. The spread of malaria is controlled by preventing the vectors from breeding and by using mosquito nets to avoid being bitten. The human defence systems include non-specific defence systems and the immune system. The non-specific defence systems of the human body will prevent pathogens from entering. A reminder that a pathogen is a microorganism that causes disease. The human body has skin, which acts as a physical barrier. The nose have hairs to trap the pathogens and prevent entry to the body. The trachea, which is also known as the windpipe, and the bronchi, the two main branches that enter into the lungs, have ciliated cells. Ciliated cells have microscopic hairs and produce mucus. This is to trap pathogens and prevent pathogens from entering the lungs. The stomach contains stomach acid, which kills bacteria. The immune system in the defence against disease involves the white blood cells. The white blood cells have three main methods to help defend against pathogens. Phagocytosis, which is the name of the process which the white blood cells engulf the pathogen. The type of white blood cell that does this are known as phagocytes. They also produce antibodies. Antibodies help fight viruses. The type of white blood cells that produce antibodies are lymphocytes. White blood cells also produce antitoxins. This helps fight the toxins that are produced by bacteria. And the type of white blood cell that does this are also lymphocytes. Vaccinations are used to help prevent infection. The immune system and its response to vaccines. A vaccination involves introducing small quantities of dead or inactive forms of a pathogen into the body to stimulate the white blood cells to produce antibodies. If the same pathogen re-enters the body, the white blood cells, also known as memory cells, will respond quickly to produce the correct antibodies preventing infection. At this point, it's key to note that the antibodies are specific to each individual pathogen. This is due to a protein on the surface of pathogens known as antigens, and antigens have a specific shape, meaning one particular antigen will have a corresponding antibody that fits it. These antibodies will remain in the bloodstream for decades. Therefore, if you are infected at a later stage with a virus or bacterial disease, those antibodies will respond much faster as they are already in the bloodstream. Vaccines prevent the spread of an infection. If a greater number of the population are vaccinated against a disease, fewer people will contract the virus. Therefore, the spread of the disease will be lower. This is known as herd immunity, where you vaccinate a population against a virus to help reduce the spread. Antibiotics and painkillers. Antibiotics, such as penicillin, are medicines that help cure bacterial diseases by killing bacteria inside the body. It is important that specific bacteria should be treated by specific antibiotics. Antibiotics are not effective against viral infections. Antibiotics cannot kill viral pathogens. This is as viruses survive inside host cells. Therefore, the antibiotic cannot access the virus. Painkillers and other medicines are used to treat the symptoms of the disease, but do not kill pathogens. Antibiotic resistance. The emergence of bacterial strains' resistance to antibiotics is of great concern. It is important to take the full course of antibiotics and not to overprescribe antibiotics, as bacteria can mutate and become resistant. The weaker strains of bacteria are killed off first when taking an antibiotic. The more resistant strains are able to survive and mutate. This means the bacteria is no longer responding to the antibiotic. The discovery and development of drugs. Traditionally, drugs were extracted from plants and microorganisms. The heart drug digitalis originated from foxgloves, which is a plant. The painkiller aspirin originated from the willow tree. Penicillin, the antibiotic, was discovered by Alexander Fleming from the penicillium mold. New medical drugs have to be tested and trialled before being used to check that they are safe and effective. New drugs are extensively tested for toxicity, efficacy, and dose. Efficacy means, will the drug be effective in killing your pathogen? The stages of drug testing include preclinical testing, and this is done in the laboratory using cells, tissues, and then live animals. This is to check that the drug is safe to use in humans. The next stage are clinical trials, using healthy volunteers first and then patients. Very low doses of the drug are given at the start of the clinical trial. This is to ensure that there are limited side effects. If the drug is found to be safe, further clinical trials are carried out to find optimum dosage for the drug. Dosage is the amount of the drug that can be given per kilogram of body weight. We have blind trials and double-blind trials. Blind trials are those in which the doctor knows whether a patient is given a placebo or the real form of the drug. A placebo is the form of a drug that does not have any active drug chemicals in it. Also can be described as a fake form of the drug. In a double-blind trial, some patients are given a placebo and others are given the real form of the drug. But neither patient nor doctor know which is the real drug and which is the placebo. And this is to remove any bias. Monoclonal antibodies are produced from a single clone of cells. Mono meaning one, clone meaning genetically identical. The antibodies are specific to one binding site on one protein antigen, and so are able to target a specific chemical or specific cells in the body. They are produced by stimulating mouse lymphocytes, these are a type of white blood cell, to make a particular antibody. The lymphocytes are combined with a particular kind of tumour cell, a cancerous cell that's able to divide, to make a cell called a hybridoma cell. The hybridoma cell can both divide and make the antibody. Single hybridoma cells are cloned to produce many identical cells that produce the same antibody. A large amount of the antibody can be collected and purified. The use of monoclonal antibodies. Monoclonal antibodies can be used for diagnosis in pregnancy tests. This is due to the urine of pregnant women containing a hormone known as HCG. Hormones are proteins. Therefore, an antibody will bind to this specific protein and be carried up a pregnancy test strip. They can also be used to measure the level of hormones and other chemicals in the blood or to detect pathogens. To research in locating or identifying specific molecules in a cell or tissue by binding them with a fluorescent dye. They can also be used to treat some diseases. For cancer, the monoclonal antibody can be bound to a radioactive substance, a toxic drug, or a chemical which stops the cells growing and dividing. Unfortunately, monoclonal antibodies create more side effects than expected. To detect and identify plant diseases. Plants may appear with stunted growth due to a nitrate deficiency. They may have spots on the leaves, indicating an infection. There may be areas of decay, growths, malformed stems or leaves, discoloration of the leaves, for example, yellowing of the leaves, indicating a magnesium deficiency, or the presence of pests. To identify a particular infection, you would refer to a manual or website, take an infected plant to a lab to identify the pathogen, or using testing kits that contain monoclonal antibodies. Plants can also defend themselves against infections. Physical defences include a cellulose cell wall, a tough waxy cuticle on the leaves, and layers of dead cells around the stems, all of which prevent the entry of pathogens. Plants may also have chemical defences, such as antibacterial chemicals or poisons to deter herbivores. Mechanical defences include thorns and hairs to deter animals, leaves which droop or curl when touched, and mimicry to trick